So one project in our lab is a project that has to do with the disruption of daily rhythms, which we call circadian rhythms. Those are your internal rhythms that are about 24 hours, like your sleep-wake cycle. And what we're interested in is how disruption of daily rhythms, which happens a lot in our normal lives, might be related to health. And in particular, we're interested in the negative health impacts of sleep loss and light at night and disruption of daily rhythms. So to look at that, we're using mice, and we house the mice in cages with running wheels, or in some cases without. And in that way, we can study the effects of exercise. So the mice without running wheels can move around their cages freely, but they're not exercising very much. The animals with wheels use them a lot and get a lot of daily exercise just voluntarily. So through this experiment, we can see if the effects of disruption of daily rhythms and disruption of sleep is maybe less if the animal's getting regular exercise. In order to study the effects of health, we are looking at the response of the animals to sugar, basically, glucose, and measuring how quickly their blood is able to clear the sugar that we give them. And we are, in some cases, giving the animals a high-fat diet, so a diet that's more similar to what a lot of us are eating, especially when we're sleep-deprived. In order to see what's going on in the brain, we use animals that have a firefly enzyme that's inserted in their genome. And so for those animals, the firefly enzyme, when the brain tissue is in culture, causes light to be emitted from the tissue. So in those animals, we can measure light coming out of the brain tissue, and it's what we call a reporter. And the light is reflecting the level of a circadian clock gene, period 2. So in that way, we're able to measure the circadian rhythms of brain tissue to better understand if there's disruption of rhythms within brain areas and if the effects of exercise are beneficial in your response to disruption of daily rhythms. Now, you might be wondering how we disrupt daily rhythms, and the way we do that is by housing the animals under a light-dark cycle that's sort of like around a really rapidly rotating planet. So instead of light and dark going through a cycle that's every 24 hours like we're used to, in our experiments, the mice live on a 20-hour light-dark cycle. And so this is way too rapid for them to synchronize to, and that's the way in which their circadian rhythms get disrupted. There's several nights where they're awake, but they're getting exposed to light, other nights when they're sleeping during the light period. And this exposure to light at all phases, all times of day, is very disruptive to the animals. Our studies are ongoing. We're not ready to tell you yet how the results are coming out, but it looks like very clearly the exercise is having a big effect on the responses we're measuring. And we're pretty excited to present our preliminary results at the Society for Neuroscience meeting this fall. This work is done by undergraduate researchers in my lab. I work at Smith College, where all the students are women, so all the work is done by women. And most of it's done by young students who are majoring in neuroscience or biology or engineering and getting ready for careers in research or health-related careers.